Day, ladies and gentlemen, today we will be discussing about culture of specialized cells, epidermis or keratinocytes. First, the isolation of samples. First of all, we obtain epidermis from a mouse and display the sample on a petri dish, which contains media, serum and antibiotic. This is to maintain the viability of keratinocytes and prevent bacterial infection. How to extract epidermis cells from mouse? First of all, the foreskin obtained from mouse is placed epidermal side down and loose connective tissue is scraped away using a scalpel. Then, the tissue is cut into smaller pieces and placed in this paste solution overnight. After the overnight incubation, the epidermis is peeled off and placed in the second dish in the medium. The end result is one dish with epidermis that can be used for keratinocyte isolation. Then, we scrape off loosely attached epidermal cells to obtain higher cell yield and wash the cells in culture medium by centrifugation and count total number of cells and the viable cells. Besides using this paste to disaggregate the skin sample, we can also use short incubation in warm trypsin or by prolonged incubation in cold trypsin. In warm trypsin, the solutions which we use are PBSA, trypsin with EDTA, medium with FBS, penicillin and streptomycin under 37 degrees Celsius. But it is time consuming and give low cloning efficiency. For cold trypsin, the solution used are PBSA, trypsin with EDTA in 4 degrees Celsius growth medium with FBS, penicillin and streptomycin. The advantages of cold trypsin is that it is non-labor intensive, high cloning efficiency and gives high cell viability. Now let's go on to the primary culture of disaggregated epidermal keratinocytes. First of all, we will seed the cells in FAD or KGM in 37 degrees Celsius. FAD is high calcium concentration medium for cultures with feeder layers. In order to culture fibroblast-free culture, we will seed cells at high density in FAD. After the cells attach, rinse with medium to eliminate non-attached dead cells. Then, continue cultivate the cells using the medium mentioned just now. This is the morphological appearance of keratinocytes after primary culture. Subculture. There are different protocols of subculturing the cells for FAD and KGM. For FAD, we need to incubate the cells in EDTA to initiate the cell detachment. Then, incubate the cells in trypsin and EDTA for 5 minutes in 37 degrees Celsius and followed by gently pipetting to completely detach the cells. In KGM, EDTA pretreatment is not required because of the low calcium ion concentration. Thus, we will incubate the cells in trypsin and EDTA for 5 minutes in 37 degrees Celsius to completely detach the cells, similar in FAD. Let's move on to the characterization of keratinocytes cultures. This is you wondering why we have to characterize the keratinocytes cultures. Cultured cells must be characterized to their epidermal or epithelial phenotype to exclude contamination by mesenchymal cells. Let me introduce you to the types of characterization methods. First, we characterize the cells by using cytokeratin-specific antibodies or the epithelial cells. Second, the analysis of expression of keratinocyte growth factors or KGF by real-time PCR. And third, in situ hybridization with cDNA. That's all for our presentation. Thank you for your attention.